Hello, my name is Albert Deglawi, and my partners are Natalie Hoyt, Catherine Tran, and Sergio Banuelos, and we're working under our mentor Demosthenes Morales III on a project for nanoparticles of protein drug delivery. So one of the major problems of current medicine is that it often has many negative side effects. Chemotherapy, for example, has many negative aspects to it. This treatment is only supposed to kill cancer cells, but it also ends up killing many other cells in the surrounding area. So the result of that is that you have many horrible effects such as bleeding, hair loss, infection, and anemia, among many others. And the reason that occurs is because this treatment, just like many other methods we use now, affect many cells and receptors and will therefore have many negative side effects.
So how can we eliminate these side effects? We believe that the solution is nanoparticles. So these nanoparticles are very small, and then they can highly concentrate any cancer cell that we wish it to go to. A nanoparticle is only the delivery system, so then we put on any drug or cargo, as we call it, and then have it go. And this interaction between the cell and the nanoparticle structure has is highly interactive, and so we change the size, composition, and structure of each nanoparticle cargo system so that it will attract to a certain cancer cell depending on the various cancers that there are. So once inside the cell, we make sure that the cargo itself will not detach from the nanoparticle until it is inside, because we are able to use a laser that will then detach the drug from the nanoparticle when we want it to.
Our contribution to this project is in the synthesis of the nanoparticle, and then in the testing to verify that we get the properties that we desire out of the particle. The first stage is taking a silver seed, which is roughly 10 to 20 nanometers in size. It's grown until we get the right size that we want to serve as our mold. We then introduce gold particles into the solution, and they have a natural affinity for the silver seed, and so they start attaching and clumping onto the surface of it. And in the end, we get a gold shell around the silver seed. And it's not a completely solid sphere, it's porous, and so it allows us to introduce chloride ions into solution. These ions will then bind to the silver inside of the nanosphere and then make them fall out of solution. So what you're left with is basically a hollow sphere that can be used in the rest of the project. At this point though, it's very small, it's about 40 nanometers in size. But it has properties that we don't desire. It's a bit unstable. It's a hollow sphere, it's very delicate. It tends to clump together and collapse on itself. And so what we do is we add a DNA complex here. And what this does is it helps to stabilize it, gives it a structural integrity, and it allows us to keep the particles from clumping together. And so that will make this complex right here. The ultimate goal is to have the protein on the end. But we can't just attach the protein to the DNA. We have to use a mediator, and in this case it's a cross-link molecule in conjunction with NTA, which is nitrilo-triacetic acid. And the way it works is the NTA uses a nickel 2+ ion, and that allows the protein to attach to the cross-link, and then this cross-link is attached to the DNA structure. So this entire structure is what we end up with. And the protein in our project is the green fluorescent protein. The property that it has is it's very easy to identify. It's a simple protein. It doesn't really do what we want it to do. It's not going to go in and kill a cancer cell, but we just use it to test it and to ensure that we're getting the protein where we want it to be.
So here we're optimizing the nanoparticle for size of the target wavelength. We need to remember the importance of catering to when and where the drug is released. So we do that by using a spectrophotometer, which measures absorbance. Here we're targeting the 800 nanometer wavelength, and we're doing this because at 800 nanometers, that's when the infrared rays can penetrate the skin without harming you. Here, we use the spectrophotometer to measure the silver seed, as you can see the blue line, which from 10 to 20 nanometers. And if you read it from left to right, it's decreasing. And as we add several increments of gold, you can see that it starts to increase, and it peaks at around 40 nanometers in diameter at 800, and we're targeting the 800 nanometer Here, you can see as you read from left to right, you can see that it's gold, and as it increases, it becomes more blue. It's kind of counterintuitive because like the silver seed is slowly, it's like gold, but it becomes more blue from 10 to 20 nanometers until it reaches 40 nanometers.
So now that we've talked about absorbance, we're going to now talk about fluorescence. And if you guys remember, this was our final structure. So now we did an experiment to verify that if we use the nickel ion, this final structure will be able to grab onto the GFP, which was the green fluorescent protein. So the experiment that we did, before we go on to that, you need to know two terms. It's quenched and dequenched. And you can think of it this way. When you heat up a metal bar, it starts glowing. When you put it inside of water, it cools down and doesn't glow anymore. We call that quenched. So when the protein is attached to this particle, it doesn't glow, and that's quenched. When it's released, it will, and that's dequenched. So the way we did this is we had our control group. The control group was this same exact structure, except it did not have the nickel ion. And what we did is we released it with an excess of GFP in the solution to give it the chance to be able to bind on to the GFP protein. And the experimental, same setup, it did have the nickel ion, and we released it also with GFP and gave it the chance to bind onto it. After we gave it enough time, we washed out the GFP and just left the particles in there. Then we added imidazole, which is a solution that was already confirmed that it causes release of the GFP once it comes in contact with the nanoparticle. And the buffer solution is just suspended. So we put these solutions inside of a fluorimeter, which will measure the fluorescence. And the numbers we got proved our theory, because these three numbers, although they vary, they're much smaller than this 387, and that's what matters. It shows that when the imidazole came into contact with this nanoparticle, there was a protein released, where these were not a high number, so that means there was not a protein for it to be released. This slide shows that our method of releasing the protein by using a laser would work, because as you can see, there's the no laser part. That shows that that sample was not exposed to laser and therefore did not have any fluorescence intensity. The imidazole, as we said, was already confirmed to release the protein at about 100%, so that has a very high fluorescence intensity. So the x-axis is the exposure time, and the color code shows the different settings of power that we used on the laser. And as you can see, the 472 milliwatt power seemed to be the best. Although this 850 milliwatt was a little bit higher, you can see it start to fall off. And that's because the protein was actually starting to get damaged because the laser was too strong. Although these are good results, we would rather these be a little bit higher and be closer to the imidazole. So there's still more testing to be done to find the optimal power and exposure time.
So this is more of a qualitative view. You're able to see your results rather than look at a chart. And we have the control cells down here on the left. And on this side, before either of them are even hit by the laser, they don't have any luminescence. So this is why we hit it with the laser to determine whether the GFP was there and then released and then cause the green glow you see on the right.
In this slide right here, we have prostate cancer cells that have been introduced with a different type of an RNA that's fluorescent in its properties. And they're taken up into the cell by chemical means. The chemical means is very effective, but it wouldn't be useful in a person. It would be toxic. And so this is just meant to serve as a control. And so this is the ultimate goal that we would like to see. We would like to see the protein inside the cell released and fully fluorescent. There are different methods that are being tested to try to increase that delivery effectiveness. And for this project, we just use the GFP, the green fluorescent protein. But the ultimate goal would also be to find a drug, a protein, or an enzyme that could help in either killing the cell by inducing apoptosis, just basically the cell killing itself, or finding an enzyme that would actually repair the internal DNA to fix whatever problems are inside. And so that would be future endeavors that could be combined with this delivery system, that you can just change the cargo that's being attached to the nanoparticle, and basically changing its target and what it does.
I would like to give our thanks to UCSB, the Jack Hancock Bridges program, the staff and resident advisors, Dr. Norbert Reich, and our mentor, Dennis T. Morales III.